think this is recording. So here's a quick overview of running peptide receptor docking with AlphaFold 2. Sorry if my voice sounds awful or if I'm kind of cough in the middle. I just got a bug on the way back home from a trip this weekend. But with that said, so we generally regard AlphaFold 2 as a valuable tool for creating PDB structures from a given sequence, but we can further leverage AlphaFold's neural networks to generate structures of docked multimer structures, and more specifically for this video, structures of peptides docked to their receptors. This example will walk through the docking of the peptide FLG22 to the FLS2 receptor. Before I jump into the practical implementation of this protocol, I just want to cover a key feature of AlphaFold 2 structure prediction model, in which these tools rely on evolutionary conserved intramolecular context to predict the structure of a given protein sequence. So AlphaFold will generate multiple sequence alignment for a given protein sequence from which intramolecular context can be inferred. And even as early as AlphaFold 2 was released, researchers have explored how these evolutionary conserved contexts can be generalized beyond intramolecular context to infer intermolecular context between different domains of multimeric proteins. Earlier examples of AlphaFold 2 took a single sequence as an input, so to get around this issue, researchers implemented glycine linkers that are generally interpreted to be flexible with disordered regions, which would give the flexibility needed for these peptides to orient itself in the appropriate orientation on the correct binding pocket. And indeed, in this paper here, they demonstrated the utility of this approach by predicting protein-peptide docking. And they were even actually able to recapitulate the conformational change in the receptor that are induced by peptide binding. Yet there's a big challenge here in that it's not broadly generalizable, as the length of the glycine linkers need to be optimized. So a later version of AlphaFold officially introduced multimer prediction capabilities. So rather than introducing a glycine linker that needed to be optimized for the binding pair, the sequences could be input into the model through a single entry with different monomers of the complex being separated by a colon symbol. Furthermore, the weights for this model were trained on multimer structures to further improve multimer prediction. So before I jump into all of the practical details, I want to mention that everything here that I cover in this video is in the lab shared drive. The documents in the drive are updated more frequently than this video. I will definitely re-record this when I'm feeling better, so it doesn't sound as bad. But the overall process and information should be the same, except for minor details here and there. So in general, stick to these documents for up-to-date information, and use this video as a helpful guide to get oriented with the material. So this protocol is largely the same as creating a monomeric protein structure with AlphaFold, and one quick disclaimer, I will be interchangeably referring to CollabFold as AlphaFold and vice versa. As discussed elsewhere in many places, the main difference between CollabFold and AlphaFold is the implementation of MMC2 to generate the MSA compared to the more time and resource intensive jackhammer that the original AlphaFold tool used. So going back on track from that aside, the protocol is largely the same except for two modifications. The first is that instead of using AlphaFold 2 PTM flag, we use AlphaFold 2 Multimer V3 flag. Furthermore, as I briefly mentioned before, to note the different domains of a multimer, we will separate the receptor sequences and the peptide sequence with a colon. It shouldn't matter the order of the sequences so long as they are separated by a colon. CollabFold is pretty user-friendly in the fact that it will automatically determine whether to implement the monomer or multimer flag based on the presence of a colon signal. So even if you forget to add this change, it should be fine. So now I'm going to go ahead and walk through an example. So please note that this tutorial assumes that you already have a conduit environment, so I'm going to show you the document I'm going to walk through. So this is also in that drive. So there are some specific examples or some specific steps you need to do to create a conduit environment because of some requisite programs you need to use. Otherwise, we're going to start the tutorial from here. So this is one quirk of my personal workflow, but I like working out of interactive desktops just because it's easier. You can just set and forget if you need to stop, and you stop in the middle of your workflow and you can come back to it and it should be saved still. In general, the same principles of it, like setting up a job in HPCC apply. So having a job under 4 hours and under helps with lowering the wait time on the queue. And also not asking for too much resources. I generally rely on 8 and 64, but you can change and modify that as needed. AMD 20 and newer is probably best, but I've had some weird things, so I always just stick to AMD 20 when I use other settings. So launch, I'm preparing this. I already set up a interactive desktop session, so clicking on this My Interactive Sessions should get my list of stuff here. And I can go ahead and launch the interactive desktop. So this is honestly just almost a virtual machine of sorts, but allows you to run stuff here. So here is the terminal. I'm going to go ahead and make this full screen. So yeah, so I'm just going to run through this. So first of all, we're just going to purge all the modules because sometimes they don't play well with each other, especially the ones that are loaded by default. And then now we're going to add conda, so module load conda 3. And then now to check if conda is loaded, we'll activate this environment command, activate local collab fold. And then once this is activated, you should have this this thing prepended to your line, showing that you are within your local environment. And in this environment, I don't remember exactly what it has. I mean, it does have Python 3.10 and a couple of things from NVIDIA. Yeah. I can double check and make sure I didn't forget any details on that instructions there. So after that, we can now add the binary, so the program to your path. And this is the one I installed in Bulging Lab, but if you run any issues, you can always install. It's fairly easy to install. So copy and paste that. And then I have some notes here. So this is broadly applicable to both monomer and multimer sequences here. So collab can take a CSV of multiple sequences, either that or an alignment file with given an A3M sequence, A3M file type, and also FASTA files. So either both a single sequence in a single FASTA file, or even multiple sequences in a FASTA file, as long as the name of the sequence is denoted with that caret symbol, or this whatever the signal is greater than signal or symbol. All right. So with that being said, I'm going to go ahead and jump onto on demand to add. Let me make sure I didn't forget anything. Yeah, so I need to add the input files into this directory in my working directory. And generally, it's best to work off the scratch because it's faster generally. The key caveat is that you do need to make sure you back up any information that you don't have. So there are a couple of tests I did before, and I'm just going to use the FASTA file that I had there before. So I'm going to copy that, and then I'm going to paste it in the working directory I created here. And then now, let me make sure I didn't forget anything. I added the conda environment, I added AF2 to the path. And one thing you can always do to check if you have that in the path, you can run local, what is it called? It's called, okay, collab fold. Collab fold batch. And then toggle the help. It may take a second sometimes. It's kind of slow sometimes. And then, yeah, and then here's an overview of all the different flags and options you can add on. And now we can just simply run this command. And then one little change here is that I added the fast and different folder here. So this is vid overview. Oh, okay. How can I copy this? One thing to note, you cannot use Command V or Command C. You have to double click or force click to get this going. And then hopefully, yep, there we go, in running collab fold 3. So this will take about 40 minutes. So I ran an example before here. And the key thing, the document always check to see how long it took is this log.txt file. And it started around 1:30, ended at around 2:10, so about 40 minutes-ish. And I think there was actually a more stringent one I did on test 1. No, I guess it's the same. But there are a couple of key features. So when you load that all in, yeah, so now let's look at the results. So this is what you'll get. So it took about 30 to 40 minutes to run this time. It depends on how long your protein sequences are and how many poses you generated. So this is for generating 5 poses or 5 different models. And here's a quick little video of that snapshot. If I can pull this up. On the right here is the peptide, I don't exactly remember. I think the dark blue is the crystal structure, and the other two are different different settings. I think the one thing difference was using templates or not using templates. And regardless of the case, they're actually pretty similar. So that is one example there. And actually, before I even jump out to the conclusion here, there's actually important things to note in terms of the output. So as I mentioned, the log.txt tells you what happened if there's any issues that the program ran into. For example, there's a small one error here that happened, but it didn't stop the program from running. And there's a couple error or a couple like accuracy metrics that you can look through. So one of these is this PAE, or predicted alignment error. And it's a picture. It gives you a measure of error for a given residue X if the predicted and actual residues are aligned on residue Y. So it's another way to think about this is it gives us a sense of the confidence in the relative position and orientation of different domains. So that's particularly useful here for multimers. Another important metric is this local per residue confidence score, PLDTT. It's scaled between 0 and 100, and 100 is the best. It gives us a sense of the accuracy of the score. So this is just a picture. If you wanted to be a little more granular about this, there's a JSON file somewhere here. One of the JSON files is a selection of score. Yeah, here we go. So PLDTT, and it's just an array of different scores for each residue. So if you need to be a little more granular with this, you can do that. And there's also tools here. So for example, I think there's a command in for the regular AlphaFold, but I think you can also color it by, I think it automatically adds a PLDTT, but I have to double check my notes on that, but don't take me on that. What else? There's one more score. Oh yeah. The predicted template modeling, or PTM score. So that gives us a more global accuracy score. And there's a modified version of that for Multimer that is the interface PTM score. And that's actually printed out when you run CollabFold, or especially in the Multimer mode, you'll get these scores. But the default, so PTM is good for a single domain, or generally, I mean, it's generated for the whole structure, but PTM gives us a more local sense of it compared to interface, like the interface of the domains. So it's a weight between the IPT, it's a weighted linear average of or linear combination of these two scores. And that's how they determine the ranking. And I think, let me see if I can pull this up relatively quick. You can, excuse me, you can modify which one or which ranking you wanted to use. But otherwise, I think that's a quick overview of how to use AlphaFold 2.